Okay, good morning, everyone. Welcome to the Centers of Excellence in Molecular Hematology, CIHD at Utah seminar series. This morning is our first of this year's 2022-23 kickoff, and Dr. Amy Medlock from the University of Georgia is going to be speaking to us this morning. I want to take 1 second. I think everybody who's on here knows this, but I'm going to take a second to, let me get there. Maybe I can get there. Share my screen. I think I'm on my laptop, so this is awesomeness. Let's see. Let me zoom down. I want to get to. See if I can get it. Okay. What do you see currently? Zoom or the? Do you see our website now?
I see your website.
Yep. That's perfect. That's exactly what I want. So this is the website that's cihd.cors.utah.edu. And if you haven't visited here, this is the Utah's U54 Center of Excellence. And across here are some drop-down menus that you can go learn about us. You can learn about things happening in the news, which are the seminar series and publications and new core activities. In here are our cores that are supported. There's an iron and heme core, a mutation generation detection core, metabolomics, and a protein metabolite discovery core, as well as our enrichment core, which is part of this seminar series. Over here, if you have a postdoc that you want to advertise, or you're looking for a postdoctoral position or other staff positions, you can come in here, and we're trying to keep that up to date, posting opening opportunities. Finally, over here in the funding link, we sponsor here at Utah a pilot and feasibility program, which was just awarded, but we do that once a year. And then if you click on the U24 link, you can go to the total Centers of Excellence sponsored by NIDDK, which has calls for pilot and feasibility projects 2 to 3 times a year, which you can go in there and learn about. Now, I just want to stop sharing and give Amy as much time as possible here. And I want to introduce her to you. So let me get one other thing here that I need. Okay, so Amy, I've known Amy for many, many years, and it's my pleasure to introduce her. She received her bachelor's degree in chemistry from, I'm going to mess this, maybe Erskine, is that right?
Erskine, yeah.
Erskine College, and her Ph.D. in biochemistry and molecular biology from the University of Georgia. She then completed a postdoctoral fellowship at the University of Cape Town, and she is currently an associate professor at the University of Georgia and the Augusta University of Georgia Medical Partnership. Work in her lab focuses on heme biosynthesis and its regulation via protein complexes in mitochondria. And today, she's going to speak to us about heme biosynthesis and distribution, complexity, and intersections. And just one other comment that I want to add, while Amy is sharing her screen, she ran a great tetrapyrroles conference this summer at Sal Regina. So congratulations on that as well.
Cool. Thank you, Diane. Thanks for the introduction. I told Diane when I started, I'm going to make that into a bigger thing, is no matter how many talks I give or how many classes I teach, I still get nervous in advance. So I'll work through that in a minute as we go, even when I'm sitting in a room by myself, that is. But so thanks for coming. Thanks for taking time to come. I'm going to tell you a little story about, or a couple of stories about how we think protein-protein complexes play a role in regulating the synthesis of heme, and also the trafficking of the intermediates in the product of heme. And so we'll come back to this model as we go through the talk, but we've done some work over the last probably 5 to 10 years showing that we see some protein complexes, and I'll walk you through and tell you how we found those and how we're kind of trying to dissect what some of these proteins do in this protein complex. So I know most of you don't need this introduction on heme, but I'm going to give it to you anyways. Heme is a necessary cofactor in most organisms make it. I'll come back to that in just a minute. But there are classical roles of heme and non-classical roles. The classical roles are those that we've known about for years in cytochromes and globins. The non-classical roles are those that have emerged in the last 10 to 15 years in terms of transcription factors, translation, regulating their circadian rhythm, protein turnover, so lots of other functions. The classical roles tend to be those where heme binds to a protein tightly and stays bound, and the non-classical roles, not always, but in some cases, are where it's part of the labile heme pool and it can be exchanged over time. About 80% of the total heme synthesized in your body is in developing erythroid cells. The second most predominant place would be liver, and then much less in other tissue. But that I wanted to emphasize that because a lot of the models that we look at are developing red cell models. There is a conservation of the complete pathway in most animals. So it's needed in most animals. Most organisms have retained the entire in the complete pathway to make heme de novo. It's thought to occur in all cells. So the dogma is that every cell makes its own heme. Any heme that leaves the cell goes to the liver for degradation. We recycle the iron. We break down the tetraperol and excrete it. An important part of the pathway is that it partitions multiple compartments. So the very first step actually happens in the mitochondria, and it's catalyzed by an enzyme called ALAS, or aminolevulinic acid synthase. And there are two isoforms of it. One that's expressed in red cells and one in all other cells. ALA is then moved out of the mitochondria where the next steps happen in the cytosol. Coproporphinogen is moved back in where it's converted to protoporphinogen in the IMS. That's imported into the matrix of the mitochondria where protoporphinogen is converted to protoporphyrin 9, and ferrochelatase, the terminal enzyme, actually inserts iron to make heme. An important component of this is that there is cytotoxicity of substrates and products. So we don't believe that substrates and products, the product heme or iron, or that the intermediates of the pathway are floating around free inside a cell. And we think that some of the regulation happens by channeling these via protein complexes, which I'll tell you about today. So some of the questions that we are looking at to answer in my lab are: how is heme biosynthesis regulated to produce heme under such different conditions seen both during normal and abnormal growth, development, and metabolism? So how is it that we can make so much heme in a developing red cell and much less in all other cells? How is this process regulated? Most of what we know is in developing red cells. We know some in the liver, but we know less in other tissue, such as the kidney or adrenal gland. And so we're interested in understanding that. How is the cellular environment actually protected from these potentially cytotoxic precursors and product of the heme biosynthetic pathway? That's another question we're interested in. And then we're also interested in what is or are the mechanisms by which heme is actually transported from the mitochondrial matrix to the different cellular locations. So we know it's made, the terminal step is in the matrix, but we've got to move it throughout the cell to other hemoproteins and the cytosol, ER proteins, nuclear proteins, and that's not quite clear how that happens. So our initial idea when we started this was that we could answer some of these questions, most likely the regulation, the protection, and the trafficking question via protein-protein interactions. And so we had a kind of naive model when we started this. I'll show you what we thought would happen and how we kind of walked through it and what we found. But we had a somewhat naive model. We knew that the terminal enzyme of the pathway had different conformations. And we knew about that via crystallographic studies, which I'll show you those conformations in a minute. But we hypothesized that these different conformations of the terminal enzyme would interact with different proteins in the mitochondria to facilitate either the binding of substrates, iron or protoporphyrin 9, or different proteins to traffic the heme out of the mitochondria so that it could be localized through the rest of the cell. So we thought that a transporter would bring in the protoporphyrin 9, and that interactions between heme biosynthetic enzymes in the mitochondria, specifically the last two, would facilitate the trafficking, the channeling of that substrate. We thought that ferrochelatase would interact with midopharin, and that was shown by Barry Pau's lab as well, so that iron could be imported and heme could be made. And then in the heme-bound conformation, that ferrochelatase would interact with the heme transporter, and the heme could leave and either via a single protein or multiple proteins be exported out of the mitochondria and distributed through the rest of the cell. And so one thing I'll say is we didn't think that ferrochelatase was floating around like this cartoon shows you. We thought that there were multiple proteins around and that we'd get these transient interactions depending on the conformation of ferrochelatase to facilitate either the substrate entry or the product release. So the approach we took to try and define some of these was we decided to use affinity purification. We flag tagged our protein ferrochelatase. We then fractionated the cells. So we did a crude fractionation in isolated mitochondria. We then lysed those mitochondria and used anti-flag bound to agarose beads and pulled it down. And then we identified the protein partners using mud pit mass spec. Our initial experiments were with murine erythroleukemia cells. And we had some nice controls in that we were using human proteins. So we use human ferrochelatase or any of the other human enzymes of the heme biosynthetic pathway. So we started with ferrochelatase, but we actually did all of the enzymes of the heme biosynthetic pathway, both the mitochondrial and the cytosolic as well. We analyzed the mass spec data, looked for things that were there in multiple experiments over time. We did reciprocal pull downs. So we tagged the protein partner we thought was there. We pulled it back out to see if we had the heme biosynthetic enzymes as well. And then we spent a good bit of time trying to characterize these interactions via both in vitro and in vivo experiments. So while we started with the MEL cells, we've actually transitioned and we're using non-erythroid cells as well. And I'll show you some of that data today as well. We've used HEK cells. We've used H295R cells, which are an adrenal cell line, and HepG2 cells, a liver cell line too. So trying to compare the protein partners in all of these cell lines to see what we see. As I mentioned, though, we started with ferrochelatase. That was kind of our wheelhouse and our area of expertise. We knew a lot about this protein. It sits at the convergence of iron and porphyrin metabolism. We assumed that a heme transporter would interact with ferrochelatase to take heme. We know a good bit about it in its catalytic cycle, and we know that the slowest step in catalysis is product release. So we reasoned that a heme chaperone would take the heme from it and speed up the reaction. And this is from in vitro studies. It functions as a homodimer. So I'm just showing you kind of a cartoon, a surface representation. The yellow molecules right here, I can actually show you, are cholate molecules. They're detergent molecules, and that would be the active site mouth. So you almost have two Pac-Man shaped molecules, and they're stuck in the inner mitochondrial membrane with their active site actually facing the inner mitochondrial membrane. The enzyme has a 2-iron, 2-sulfur cluster, 1 per monomer. So you have this kind of Pac-Man shaped molecule with an iron sulfur cluster. This iron sulfur cluster is required for activity, but it doesn't play a role in catalysis. So it doesn't donate.
Oh, sorry. I just logged into a different meeting. Sorry. Yeah. I'm listening.
So we do know from our crystallization studies, though, that it seems to be a dynamic protein. And I'm going to show you that in the next video as well. So we solved structures of a number of different variants of ferrochelatase. Some had substrate bound, some had product bound, and I'll walk you through this in a minute. And from those, we made a movie based on those crystal structures. And what it looks like occurs during the catalytic cycle is that porphyrin binds and our Pac-Man shaped molecule closes down and completely engulfs the porphyrin. So that'll restart in a minute. So here's our porphyrin entering the active site. The active site mouth closes down, completely engulfing it. The iron is then inserted. And we have a conserved pi helix in each subunit that partially unwinds, making an alternate kind of release conformation for our heme product, and our heme leaves, and we think in a possibly different way than the porphyrin enters. And that would be mediated by protein-protein interactions. We know a little bit more about this enzyme from studying the very first enzyme in the pathway, which I'll relate it back to in a minute, but we think that ferrochelatase is a second regulatory point. The rate limiting step, or the main regulatory point in the heme biosynthetic pathway, is the first step, but we think that ferrochelatase is also important when ALAS levels are in excess. So when it is no longer rate limiting, ferrochelatase seems to be rate limiting as well. Based on these different conformations and what I showed you already, that led us to postulate that there would be different protein partners. Before I tell you about what we found, though, I do want to tell you that one of the things we noticed early on is when we express ferrochelatase or overexpress ferrochelatase in our cell lines, that we didn't see much of a difference in heme levels. We got a broad range in our heme levels. And again, these are murine erythroleukemia cells, so they make a significant amount of heme. You can induce them, but these were in undifferentiated cells. So the heme levels, there was no significant difference, but what we did see was that the porphyrin synthesis was actually altered in these cells. So we had an excess of our porphyrin precursors, suggesting that ferrochelatase may in some way stimulate early steps in the pathway to increase the amount of porphyrin precursors. So we didn't get excess protoporphyrin in these cells. We did see elevated zinc protoporphyrin and then the porphyrin precursors as well. So when we looked at these enzymes in cells for protein-protein interactions, one of the most interesting things, and I put this in here for John Phillips, who's traveling today and not on, is that we saw no interactions in the cytosolic enzymes. We looked at all the cytosolic enzymes and reasoned that they would form a super complex to channel substrate in the cytosol, and we saw nothing. But what we did see in the mitochondria is that the terminal two enzymes, ferrochelatase and protoporphinogen oxidase, actually interacted with the very first enzyme in the pathway. And that was a surprise. We had hypothesized that protoporphinogen oxidase and ferrochelatase would interact to channel substrate so that it wasn't free in the matrix, but it hadn't been proposed that it would interact with the first enzyme in the pathway. And this was in the murine erythroleukemia cells. We've subsequently repeated this, and we see that interaction with the non-erythroid form of ALAS1. So in non-erythroid cells, this seems to hold true as well. We haven't gone back to measure porphyrin precursors in these cells yet, but that's something on our list to do. So we think that the increase in precursors could be occurring via the interactions with ALAS1. That may not be the only way, which I'll show you how we kind of look to characterize that. So since they interacted, one of the easiest questions we thought to ask is, how do these proteins in vivo affect each other's activity? And so we did some assays of ferrochelatase in which we have just ferrochelatase, or we added purified ALAS2. Ferrochelatase, this is its activity. ALAS2 alone has no ferrochelatase activity, obviously. And if we included it in the assay with ferrochelatase, we had about the same amount of activity. So there was really no change in fetch activity with ALAS present. Interestingly, though, when we look at ALAS activity in vitro, here is ALAS, here's fetch. And then if we included fetch in the ALAS assay, there was a small increase in activity. Not a lot, but there was a small increase in activity. And that may partially explain these porphyrin precursors that we see. Another thing that was happening about the same time in Harry Daly's lab is he was looking at where the precursors for ALA production come from. So I know I'm kind of jumping around a little bit at the beginning and the end, but if you remember, and maybe I didn't, I think I did say this early on, but anyways, the precursors for heme are succinyl-CoA and glycine. So that's how you make the first the first intermediate in the heme biosynthetic pathway, ALA, is from succinyl and glycine. And so the dogma that kind of had existed was that succinyl-CoA was being pulled from the citric acid cycle and being utilized to make ALA and thus heme in most cells. That seems plausible if you're making a small amount of heme in a normal cell and not an erythroid cell. But if you're making it in an erythroid cell where you need a lot of heme, a significant amount of heme, you would be pulling tons of succinyl-CoA out of the citric acid cycle, depleting it and essentially stopping the flux through the TCA cycle. So work in Harry Daly's lab with Joseph Birch used some labeling studies, and in these studies he showed that glutamine via glutamate is actually the carbon source for porphyrin production in the erythroid cells. So we're not pulling succinyl-CoA when we need huge amounts of it out of the TCA cycle. Instead, we are using glutamine via glutamate via alpha-ketoglutarate dehydrogenase right here, and that's how we're getting the succinyl-CoA for ALAS. And so we looked back at our data and our pull down data for ferrochelatase and for ALAS2 to see if we saw interactions with KDH in these cells. And we do. We see ALAS and ferrochelatase, they actually interact with alpha-ketoglutarate dehydrogenase, likely not in a super molecular complex of the TCA, but in a moonlighting role pulled out of the TCA cycle interacting with these enzymes. And this is another mechanism by which we could explain this increased porphyrin production in these cells. So both by stimulating ALAS2 activity, but also by making a complex to channel succinyl-CoA for ALAS or ALA synthesis, sorry. So those are the mechanism. So our model from this is that we have a heme biosynthetic pathway, or the heme biosynthetic pathway enzymes form a metabolon in the mitochondria. And we think that this regulates heme synthesis via substrate production. So via the production of these intermediates that are going to leave the mitochondria, be further metabolized in the cytosol before they return, and in the delivery of substrates once they re-enter the mitochondria to make heme overall. So in the mitochondria, we think this metabolon is really important for regulating both the intermediate production and channeling the substrate as it re-enters.
Amy?
Okay. Yep.
This is Diane. That channeling in and out of the mitochondria, is it, we still don't know what's moving it in and out, correct?
No, yeah. I don't think we do know how glycine's getting in, but we don't know how ALA is getting out.
Right, and it looks like Sarah, do you want to ask your question? Go ahead.
Yes, hi, Amy. You remember how ALAS2 has CP domain for heme fixation, and no one really know what they are used for. Do you think that they are important for this link between FEC and ALAS2 and FEC modulating ALAS2 activity? You're mute, Amy, sorry.
Let me unmute. Okay. Yeah, possibly. That could be. We haven't looked into that though, Sarah, to see if that's the case. But yeah, we do have some data though that I could go back and look at. We did some pull downs with some of the XLPP variants of ALAS2. So I could actually go back and see if there's some differences there.
Sorry, the CP domain, I think, are a bit different from the C-terminal domain of ALAS2.
Oh, okay.
It's like in ALAS1, they are supposed to modify like the translation, the move of the enzyme to the mitochondria and everything. But in ALAS2, they are there, and no one really know what they are used for.
Yes. Sorry, I misunderstood. I thought you were talking about the C-terminal domain. No, yeah, we haven't looked at that, but that is a possibility that those could mediate that. Okay, cool.
Thanks.
Any other questions?
I don't see any.
Okay. I'm going to move to kind of the next part. And this was something that, we noticed in our original pull down data in our metabolon, and then in collaboration with Ole Kalamanchuk and Amit Reddy at Georgia Tech, we investigated this a little bit farther. So what we saw and Ole saw as well is that the heme biosynthesis enzymes, specifically we saw the heme biosynthesis metabolon proteins interacted with the mucose or the mitochondrial organization system proteins, and also some proteins involved in mitochondrial dynamics. So we saw those in our original pull downs in our erythroid cells, specifically a protein called IMMT, which is the homolog of MYC60, and OPA1, which is the yeast homolog of MGM1. So we noticed those both in our erythroid cells. Subsequently, we've done non-erythroid cells for ferrochelatase, and we see those proteins are there as well as other proteins of the mucose complex. Ole did pull downs in yeast, where he tagged Heme15, which is yeast ferrochelatase, and pulled it out. He pulled out many of the same proteins that we saw in the human cells and those involved in mitochondrial dynamics as well. The mucose complex organizes mitochondrial structure, so it's important in those cristae junction points, junction between the inner and outer membrane. And we also know, or from previous studies, that proteins involved in mitochondrial dynamics interact with the mucose proteins as well. So Ole and his lab did a lot of work with the yeast knockout strain of MYC60, and he noticed some interesting things early on that with Amit Reddy and some work in our lab looking at enzyme activity, he investigated a little bit farther. So the first thing he saw was that MYC60 yeast cells were respiratory deficient when you express ferrochelatase. So over here in this panel, if we express ferrochelatase under a low expression promoter or a high expression promoter, these cells were respiratory deficient. We looked at the level of fetch in these cells, the wild type and the knockout, and we didn't notice a big difference in the amount of protein in these cells. But when we actually measured the activity of ferrochelatase in these cells, what we found was that ferrochelatase activity was decreased in the MYC60 knockout. So for some reason, we seem to be making them sicker by overexpressing ferrochelatase. They were respiratory deficient. They had similar levels of FEC protein, but that FEC activity was decreased overall. And you can see that right here as opposed to the wild type cells. So we questioned if the cells were iron or heme deficient, or there were other problems in the heme biosynthetic pathway that would help to explain the respiratory deficiency that we were seeing in the decreased ferrochelatase activity. And so Ole, in collaboration with Amit Reddy and also the Iron and Heme Center at the University of Utah, found that heme and iron levels in the MYC60 cells were similar. So he didn't see a big difference in those overall. But what he did find was that intermediate porphyrin levels were increased in the MYC60 cells. So both the MYC60 cells without ferrochelatase expression and with ferrochelatase expression. And then he found that if you overexpress ferrochelatase in the MYC60 cells, they had lower levels of protoporphyrin 9. These, I do need to say, these intermediate porphyrins that we're looking at are the oxidation product of the porphinogens, which are intermediates of the heme biosynthetic pathway. So these intermediate porphyrins are not intermediates. The reduced form, the porphinogens are, but we're measuring the oxidized form there. So Ole in his lab went on to look at a little bit more, trying to characterize these. They seem to have increased levels of porphyrin. We weren't able to specifically figure out where those porphyrins were, if they were in the cytosol or the mitochondria. But what Ole did do in his lab is find that there was increased oxidative stress in the MYC60 cells when they were expressing FEC. So he found a decreased aconitase activity and hydrogen peroxide survival and growth, and that this could be partially rescued by including in acetylcysteine, the antioxidant. So he could get at least partial rescue if he decreased that oxidative stress in the cells. The other really clever thing that Ole and Jonathan did in the lab is that they made a synthetic tether, and they were able to rejoin the inner and outer membrane of the mitochondria with this synthetic tether, MitoT. And by doing so, they could they could partially rescue the growth or the respiratory defect by synthetically connecting these two membranes. And so our model that we have right now is that the MYC60 is going to be necessary for the import of the protoporphinogens to the mitochondrial matrix. We think that it's helping to position the metabolon at these inner outer membrane junction points, and that's going to be necessary for facilitating the import of the porphinogens into the mitochondrial matrix. So when mucose is present, the porphinogens are imported in and able to be utilized by ferrochelatase. When they're not, when mucose is absent, we get an accumulation of the porphyrin precursors outside the cell, and we get oxidative damage, most likely outside and inside the mitochondria as well. We don't see a decrease in heme, so we must be able to get some of these porphinogens back in some other way to be able to have heme levels, because if you remember early on, there was not a decrease in heme or iron levels overall. So we think this is kind of our model now, that the mucose complex and the metabolon is positioned near the mucose complex to facilitate both the import of the porphinogens and the export of heme. All right, I'm going to switch gears one more time and tell you about another protein in this complex. And this protein is an interesting one. It's one that we found early on. I'll stop. There's a question before we go on.
Hey, Amy, great talk
hey.
so far. Good to see you. I had a quick question. Maybe it's a naive one, but did you also look in, maybe in this case, they're recycling or scavenging heme and not bringing in the porphinogens? Like, have you tried any of Amit's, forget what they're called now, but the labile reporters, and look at cytosol versus mitochondria there?
We did. We used Heme sensors to measure heme levels. I can't remember, Ole may remember, did we look at trafficking though? That's what you're asking. So could we get trafficking? I can't remember if we looked at trafficking in these cells or not, or just looked at total heme levels with the sensors.
Yeah. Hi, this is Ole. Yeah, we did. And it doesn't really change that dramatically. So, yep.
Cool.
Yeah.
Thanks. Good to see you.
All right. Any other questions in terms of the localization? All right, I'm going to switch gears and talk about one more protein. And this protein has been a thorn in my side for a very long time, as has it for many other people, I will say. And I've listed some of them here on the slide, some of them which are affiliated with the University of Utah or the center who've worked on this protein over the years, including Amit Reddy and Adam Hughes, Peter Espenshea. There's a whole group of people. So when we originally did these studies, we found a small protein called progesterone receptor membrane component 1, or PGRMC1 for short. This protein is a bit of a mess in the literature. There's, I don't know how many papers, 400 or 500, probably even more. The only thing that's readily agreed upon with this protein for the most part is that it binds heme. That's about it. There's one other thing that I'll say is fairly agreed upon with it, but that it binds heme. It may possibly also bind progesterone, although that has been argued over time. It seems to bind progesterone, and it seems to influence the heme binding as well, or the heme binding site as well, when it does bind progesterone. The other thing that's fairly well established and agreed upon is that it's known to stimulate some cytochrome P450s. There are quite a few, I think there's up to 20 now, that it's been shown to interact with and stimulate their, or alter their activity. Some stimulate, but in general stimulate. It has many other reported cellular functions. You want to give me one, it probably has been reported to do that. It's increase in cancer cells. It's been reported to modulate growth factor receptor. Did I say fatty acid metabolism? You name it. It's been reported to do just about everything over time. It's also been reported to be localized all over the cell. It's agreed upon that you can find it in the ER readily. We seem to find it in the mitochondria, although I'll argue that in just a minute with you. But it's also been shown to be on the cell surface or the nucleus. So you name it, it's been shown to be all over cells. I put this in because people always ask me about knockout animals. Have there been knockout animals of this? There have been some zebrafish, and they mainly looked at reproduction. So there's kind of two fields that seem to be working on this protein. One is in reproduction. Of the knockout mice, one of the knockout mice lines showed steatosis on a high-fat diet in cardiac tissue in the liver, which was interesting. Another one showed protection from acetaminophen toxicity and regulation via cytochrome P450s. And both, this was a more recent one from Peter Espenshade's lab. Interestingly, if you look at the CBC of these mice and the whole body knockout, they are not classified as anemic. They're within normal parameters, but you see a decrease in the red blood cell count, the hematocrit, and the hemoglobin, about a 10% decrease in these animals when you knock them out. But they're overall generally okay. They can reproduce and they seem to be okay overall. This protein, just to kind of give you a little bit more background, another thing people always ask about, well, is there a structure? Can we gain anything from the structure, understanding the structure and how it binds heme? And there is a structure, a crystal structure of this protein. It's shown right here. The structure, though, is only of the soluble domain. So it has a transmembrane domain and a soluble domain. And so the crystal structure is just of the soluble domain. And this crystal structure shows two heme stacking. And so it's a little bit hard to see here, but here is one subunit in aqua, here's one subunit in green, and then there are two hemes, and you notice they're quite exposed, right? And that they are stacked as well. I will say that the way that they got heme bound to this protein in the crystal structure was by adding heme to the protein, or actually to the crystals, I think. And heme is known to stack at high concentrations. So it's not quite clear if the heme actually is stacking within the PGRMC1, or this may be an artifact of high heme. We played with this a little bit more based on the alpha fold structure. And so I want to show you, we took that alpha fold structure, and it's of the full length protein. So, and we actually dimerized it. It's known to function or to be a dimer. And so we actually dimerized it and looked at it overlaid on top of the actual crystal structure in the biological subunit. And so this is what it looks like. Sorry, the colors aren't great, but it's in this darker blue color and this kind of olive green color right here. And the first thing, if I go back and forth, you can see that this area right here is the transmembrane domain. So one of the subunits in the crystal structure is actually where the transmembrane domain and a dimerization interface would be predicted. So you can kind of go back and forth. So based on the alpha fold structure and overlaying it with one of the subunits and one of the, or both of the hemes, if we actually look at it, it looks a little bit more like one heme may bind per dimer. We're not quite sure, and we're trying to tease this apart by making some mutants and looking at oligomeric structure of these, but it almost looks like it fits a little bit better. You can see the second heme would be in this second monomer right here, but it looks like it might be right there. And I have a movie because I think it's easier to see with a movie. Let me do this so you can see it kind of rotating. We do know that it binds ferric heme better than ferrous heme. So that's an important point. It has one active site ligand. It's a tyrosine. You can see it right there. And there's another tyrosine in the blue one that's just behind it. And they're both positioned so that they would be kind of right at the iron. So it almost looks like maybe the biological unit or the dimer may be actually binding a heme together, but we're not quite sure. We've also done some studies where we dimerized via the alpha fold, the oligomer prediction with PGRMC2, and it actually looks quite similar to the PGRMC1 dimer. So making a heterodimer, because we know that PGRMC1 can interact with another protein called PGRMC2, which is very similar, which I didn't really tell you about, but I'll come back to in just a minute. But that's kind of what we were thinking in this structure. And again, we're investigating that a little bit farther. So from our initial affinity purification experiments, we showed that fetch and PGRMC1 interact, and these were in our erythroid cell line, but we've subsequently showed this in other cells. We verified this with reciprocal affinity purification. So we tagged PGRMC1 and PGRMC2, pulled ferrochelatase back out. We again validated it in a non-erythroid cell line. I was very nervous about it because it's overexpressed in cancer cells, and we have a erythroid leukemia cell line, and so I wanted to make sure it was true in other cell lines, so we were able to do that. We were able to show that PGRMC1 and PGRMC2 interact. And I'll come back to that in just a minute because there's been some work done on that suggesting it's a heme chaperone. And then PGRMC1 actually interact with other metabolite proteins as well. So we saw it with protoporphinogen oxidase and some of the other proteins that we saw in the complex overall.
So
We
Amy,
did. Yep.
this is Diane.
Yeah.
Is the knockout show deficiency in heme containing proteins at all? I mean, I know you said the red blood cell content is a little bit
Yep.
lower, but I'm wondering about the activity of, catalase or any other heme containing enzyme that you could measure.
Not that they know of. They did show, again, if you knock it out, it was protective from acetaminophen toxicity. And this is a little counterintuitive, but acetaminophen is metabolized by a cytochrome P450 to a toxic byproduct called NAPQ that causes liver damage. And so if you don't have PGRMC1, it doesn't get metabolized by that cytochrome P450. So that would suggest that that cytochrome P450 is not active, possibly because it doesn't have its heme. But there's no global phenotype that you can see of heme deficiency overall in these animals, no. They
Amy,
they also had to stress the animal to see anything. And you notice that from the knockout studies that I mentioned, they had to put them on a high-fat diet or they had to treat them with acetaminophen. Yep.
Amy, I was going to save it for the end, but since Diane jumped in, that also follows up to my next question, which is you mentioned reproduction as well.
Yes.
I only bring it up because from my work, I know that sometimes you don't see an overt hematological phenotype, but indeed it was in the testes where we saw the issue manifest with the multi-drug resistant proteins, the heme transporters. What was the issue with the reproduction in the PGMRC1 knockout?
You know, I have to go back and look at all that. I don't remember all of them. I know in the zebrafish, they noticed that there were slight problems in reproduction, but I can't remember all the details of those. I don't think they saw any reproductive issues in the whole body knockouts of the mice. There are some groups that have tried to knock it out of other specific cells. I think granuloma cells, and I can't remember the others. And I can't remember what that knockout is, but it, you know, in the whole body, at least it's not that big. Okay, so we jump back to some in vitro experiments, and what we first did was look at its effect on ferrochelatase activity. We know they interact, and I'll show you that we showed that they interacted in vitro as well. So we hypothesize it's a direct interaction, although that's a little hard to reason through considering the subcellular location, but we'll come back to that in just a minute. But ferrochelatase, we found that ferrochelatase activity decreases in the presence of PGRMC1 by about 40%. So we titrated in PGRMC1 and looked at fetch activity overall. Because we have different conformations of ferrochelatase, by having these different variants. So either our open conformation, which is our wild type that can accept substrate, our substrate bound conformation, which is E343K, or our heme bound conformation, which is the F110A, we found that PGRMC1 interacts more tightly with the heme bound form of ferrochelatase. And that was pretty exciting because we thought, it's interacting with the heme bound form. Maybe its role is to take heme away. But with that, we were a little confused with our assay experiments as well, that we thought it would actually stimulate activity if we're taking away, but maybe we were saturating the PGRMC1. I will give you another, just aside, a problem that we've had dealing with it is it co-purifies from E. coli with some heme bound. So you get anywhere from probably 30 to 40% heme bound. We haven't been able to make it fully apo or fully holo either. We haven't been able to load it all the way or to strip it all the way. And because of that, that's led us into problems in terms of looking at affinity for ferrochelatase, because we get some weird experiments. We tried a lot of different ways to look at its interactions with ferrochelatase, but we haven't been able to do so. We also have, and I'll just throw this out there, we've tried co-crystallizing. We can readily crystallize ferrochelatase in multiple conformations. We tried to co-crystallize full-length PGRMC1 with ferrochelatase, thinking that might be kind of the magic. We weren't able to crystallize the full-length protein on its own, nor with ferrochelatase, unfortunately. And we've tried that several times. So this for Kevin Hicks. So we decided that we'd send Kevin our PGRMC1 protein, and he could use it in his MIDA screen to look at metabolites that might interact with it to give us some clues about what it's doing. And so these are the first pass results, and Kevin's looking at this protein again shortly. But he's found 6 proteins that have a positive fold change, indicating likely a non-covalent interaction. And then he had 5 proteins with a negative fold change, indicating a higher affinity interaction. And what we noted from these, kind of the take home, we still don't really know what they mean, is that we clearly see a glycerol 3-phosphate and then a purine nucleoside or nucleotide signature in many of these molecules. So trying to figure out maybe if they're regulating it or playing a role in it. We went back to some affinity purification, and we looked in cells that are not MEL cells. So here's our MEL cells. We looked in HepG2 cells and HEK cells as well. You get a large number of proteins from HEK as compared to the MEL and the HepG2 cells. What we found when we looked at all those proteins, especially the ones that we see overlap between 2 or 3 cell lines, most of the protein partners are either mitochondrial or ER proteins. So this is just showing you a graph of what we found. And that when we looked at pathway enrichment of these proteins overall, we see enrichment of mitochondrial and ER processes. And so I highlighted a couple of these for you. cristae formation was one, cholesterol biosynthesis, which has been reported for PGRMC1 before, heme biosynthesis, metabolism of lipids, mitochondrial or cytosolic calcium levels, and then phospholipid metabolism as well. Ole then helped us look at a little bit closer in more purified fractions. What is the subcellular localization of PGRMC1? We originally localized it to the mitochondria, specifically the outer membrane from some work we did previously. But Ole did more stringent purification processes, and based on multiple proteomic studies that had identified PGRMC1 as existing at ER mitocontact sites in mammalian cells, Ole carried out fractionation in rat cells that's consistent with PGRMC1 localizing to MAMs. So what we actually think is happening is that it's tethering, or it's important in that interaction between mitochondria and ER. We then worked with Amit Reddy and went to the yeast system so that we could use his trafficking assay and his heme sensor. And we looked at the yeast homolog, which is called DAP1, and its role in heme trafficking. The first thing that Amit did was show that DAP1 and human PGRMC1, because they're a bit different in terms of their amino terminus, can both rescue the DAP1 yeast strain. And then when he did his trafficking assays, what he found was that DAP1 deficient yeast cells that don't have DAP1 are deficient in nuclear heme trafficking. So cytosolic was pretty much the same with the wild type and the knockout cells, either with empty vector or with the rescue of DAP1 or PGRMC1, as were mitochondrial trafficking. But nuclear trafficking was severely affected when he had the DAP1 knockout in our empty vector, and he could rescue that with either the yeast protein or the human protein. We have not tried to rescue it yet with PGRMC2. But he is working on that to see if that would actually rescue it. We turn back to our erythroid cell line, and we use CRISPR targeting to disrupt PGRMC1 and K562 cells. These are the human equivalent of the MEL cells. Interestingly, what we ended up finding was that there was an increase in the total heme in both the undifferentiated and the differentiated K562 cells. So here are undifferentiated and our differentiated, our wild type versus our knockout cell line. We looked a little bit closer via the Iron and Heme Center at the University of Utah. And what we ended up finding was that there was an increase in the total heme. We also had an increase in porphyrins and an increase in zinc protoporphyrin as well in these cells. We then sent the cells to James Cox at University of Utah, and he did metabolomic analysis on them to look for metabolites that were different in our wild type, which is shown here, and our knockouts, which are shown here. We see over 30 different metabolites that are different in our cell lines, either our wild type or our knockout. And some of those are related to MAM processes, things like phospholipid synthesis, lipid metabolism, and single carbon metabolism. So what we think we're looking at is a model, and this is not our model. This is a model from the folks who did the work on PGRMC2, that PGRMC1 is going to be important for the mito-ER junction points, and that we are going to traffic heme from the mitochondria to the ER to PGRMC1, which will, the trafficking then goes to the nucleus, and this is work that they've done showing that it's involved in nuclear heme trafficking. So we think it's going to regulate the heme synthesis via these mito-ER junction points. We think it may function as both a sensor and a chaperone, possibly, because we know that it regulates ferrochelatase activity. And then there's some other, besides the work we've done, there's other support out there that suggests that this might be the case, that PGRMC1 may be at these mito-ER junction points. So our model, sorry, I think I didn't talk too fast. I thought I would talk a lot faster. So our model right now, it looks like this. So we've added to it a little bit. We have an ER kind of ish structure right here. So the heme synthesis pathway enzymes, we think, form a metabolon. We think that it's important that it's at the inner outer membrane junction points, regulated by the mucose complex, and that it's also got to be, or it's important for it to be at these ER mitocontact sites to regulate heme synthesis and trafficking to the nucleus and overall trafficking and regulation of the pathway. So that's kind of the end of it. Most of this work was done by David Obie, who's a grad student in my lab, or Robert Peel, who was a grad student and a postdoc in his lab. Had some great technicians and research scientists who worked on this, and also this amazing high school student who's now out in California. This is all done in collaboration with Harry Daly at UGA and his grad students, Jason, Sophia. Tammy helped a lot with the original work we did, and Joseph Birch. We have a lot of awesome collaborators at the University of Utah, at Georgia Tech, at Nebraska. All the mass spec, I do need to say, was done at UCLA by James Waschlegel. And then Barry Paw started a lot of this work, who was at Harvard. We have another project that I didn't have a chance to talk about today that we're just starting via a pilot and feasibility grant that's with Beverly at the Hutch, and Brian and Dan have been essential in that. And that's another project where we're looking at regulation of heme biosynthesis, but in this case by an immunometabolite. And I'll hopefully have some data to tell you guys about that again soon. So with that, I'd be happy to answer any questions that I haven't answered yet.
Awesome, Amy. Thank you so much. Ian, I see you're the first hand up, so go for it.
Yeah, maybe it's just from deja vu, but great talk, Amy. And I just had a quick comment / question for you and Ole, especially seeing the MAM subcellular localization, right? That if you had talked to Iqbal at all about my PNAS paper, but if indeed it's possible, it's handing it off to the MRPs in order to do the subcellular trafficking. And if in the pull downs, have you seen ABCC5 or ABCC12?
I don't know. I'd have to look back at that. ABCC5.
Yeah, the MRP5, MRP5
Yep.
or MRP9, ABCC.
Okay.
It's my PNAS paper from last year.
Okay.
But
Yeah,
talk to Iqbal. I'm not there anymore, but I think it's very intriguing. It's like deja vu.
Yeah, yeah.
And we see very similar stuff with metabolites and the changes in them in the subcellular fractionation.
Yeah, I'll have to look back and see.
Cool.
Cool.
Okay,
Great talk.
Carolyn
Thanks.
Philpott.
Amy,
Yeah.
thank you. That was an interesting talk. Towards the beginning, you were showing us your structures of ferrochelatase, and you mentioned that the iron sulfur clusters were 2 iron, 2 sulfur clusters.
Yes.
And, from, way back when, the dogma had been, that there were 4 iron, 4 sulfur clusters. How did you come to understand that they were 2 iron, 2 sulfur clusters?
Not for ferrochelatase. They've always been, I think, identified as a 2 iron, 2 sulfur cluster. In the mouse and the human, all that initial work, they're always been. You're not thinking of PBGS, are you?
No. I
No,
mean,
no?
well, maybe I've just been wrong forever, but the clusters have always been 2 iron, 2 sulfur clusters.
As far as I know, Harry can say, but as far as I know, they've always been known to be 2 iron. Yeah.
They've always been.
Okay. Well, that's helpful. Thank you.
I totally had that wrong too. That was one of my questions. Thanks, Carolyn, for asking it.
Olay, did you have a question
No, I didn't. I was
or a
just,
comment?
I was just going to say, if someone's interested in these subcellular fractionations, we're happy to help. I mean, we've been isolating MEMS routinely. Yep. Just to clarify the mysteries.
I'm going to apologize for the background noise, but I have one other question, only based upon ABCB10, of course. Do you guys in any of your crystal structures see heme breakdown products like biliverdin or bilirubin?
No.
I'm trying to
We haven't.
understand that as a transporter and why you would see those breakdown products in the mitochondria. Has anybody else measured or seen bilirubin or biliverdin in the mitochondria? Doesn't look like it. Okay. Other questions? Anybody? Go ahead and unmute yourself and chime in. It was a great talk, Amy. Thank you so much.
Thanks for the invitation.
Is it okay to ask a really unrelated question, but to the group, because I'm just seeking help? I was looking for zebrafish PCR primers, iron related. If anybody has some zebrafish PCR primer list they'd be willing to share, well, I'm just gonna put my email in the chat. And I would just greatly appreciate it if that. So new into zebrafish, and we're just kind of getting started, and appreciate anything we can do to get the ball rolling.
Mikhail, if you reach out to me with an email, I will connect you with Yvette Yen, who is not on this.
Okay.
She was trained in Barry Paz lab, and she is a zebrafish expert and can help you for sure.
All right, that would be fantastic. Yeah, it's already hard enough starting from scratch. We got to start breeding those little guys now. So just trying to save time. So,
and we have a couple of lines that you might be interested in, Mikhail, too. So let me know. We have the beta globin GFP from Linzon. We have the ferrochelatase knockout cell line that was from one of the big screens, and then we also have an OPA1 knockout line from one of Olay's colleagues that we met. So we have a couple of different lines if you'd like to. Have some.
Yeah, great, thank you.
Okay, well, we're just at 10 o'clock right now. I want to thank everybody for attending and want to make sure I invite you, to our next talk, which will be given by Sarah Ducamp on November 8th. And she will be talking about factors modifying the severity of erythroid protoporphyria. And so I hope we'll see you again. And again, thanks to everybody for attending, and especially thanks to Amy for all your efforts and presentation and taking on this first talk for this seminar series. Thank you so much.
Thanks.
Great job, Amy.
Thanks.
Everybody take care.
Have fun on your vacation, Diane.
I will. Thank you.
Yeah, thanks.
Hey, Amy, stay on.